Why you didn't take this lesson? Lesson 13.3. Mutation. Any heritable change is called mutation. Mean change in the DNA or RNA is called what? Change in the DNA or RNA is called mutation. Now, if you look to mutation, mutation is what? It's actually the change. Change in genetic information. Right? With the passage of time, change occurs in the organism. That is due to what? That is due to mutation. Mutation can be beneficial or it can be harmful. Right? If it is beneficial, so mean it can help us. It can help the plant. It can help the animal. Right? But if it is harmful, it can cause diseases. Right? Like thalassemia, you know? Thalassemia, hemophilia, osteogenesis imperfecta, color blindness. These all are diseases which is caused by the mutations, genetic mutation. And we call these diseases genetic diseases, right? So if you look to genetic diseases, it cannot be treated easily, right? It can take several hundred years. It can take several hundred years. For example, if a disease, a genetic disease start in a family, so it will travel. It's a genetic disease. Now it's a part of the DNA. So if it is a part of the DNA, the disease is a part of the DNA. So if it is a part of the DNA, so mean it will go in the next generation, and so on. So treatment will take generations, 2-3 generations. So its treatment is not easy. If you look to the types of the mutations, so mutation has two basic types. Basically, it's divided into two types. One is chromosomal mutation and other is gene mutation. Gene mutation is also called point mutation. Sometimes we are saying it said three types: genes, point, and chromosomal. But gene mutation and point mutation, that is the same type, right? Then gene mutation is divided further into three types, and chromosomal mutation is divided into four types. Right? Gene mutation, substitution, replacement, substitution, addition, and deletion. These three types, right? This occurs in the genes. In one gene, like for example, you have the book, right? In book you have 13 chapters. This book is take the example of book as the entire genome of a person, like entire human being, entire plant, entire animal, right? But change occur only, let's suppose, in chapter 13 on page 12. Right? Let's suppose change occur only on one page. So this is what? This is gene mutations. Only a slight change, little change. Now it has three types. Yes. Deletion, insertion, and substitution. This we studied. Next one we have chromosomal mutation. So chromosomal mutation means the change occur in the entire bone, right? Or many places in the entire bone. So it is a large amount of change, right? So if there is a large amount of change, so what will happen? It will have more chances to cause disease, right? If you look in the gene mutation, so gene mutation, the first type which we studied, replacement or substitution, right? Replacement or substitution, it do not have effect or very less effect. But the rest of the two types, it has effect, right? So either good or either bad. And if we talk about the chromosomal mutation, so almost it all have effect. It will be either positive effect or negative effect, right? So that is about mutation. Now next we are going to study that is gene. The next lesson which we are going to take, that is gene regulation and expression, right? Gene regulation and expression. If you remember till grade 9, you were saying in biology, what was the smallest thing? The basic structure and function unit of life was what? Cell in biology, cell, right? Cell is what? Entire living things are made from what? From cells. Right? So we are saying that cell is the basic structure and function unit of life. That was till grade 9. When you reach to grade 10 or at the end of grade 9, you say nucleus inside the cell, right? Nucleus inside the cell. Now from chapter number 10 in grade 10, right? From chapter number 10, you studied DNA and RNA. Where DNA and RNA is present? In the nucleus, right? It is present in the nucleus. So mean from the cell you enter inside the nucleus. From the nucleus you enter inside the DNA then, right? Now from the last lesson you are saying mutation, gene mutation or chromosomal mutation. Now chromosomes and gene, it is present in DNA, or DNA made the genes and chromosomes. Now, if you look the entire function of the body, the entire function of the body, the entire function of an organism, it is controlled by the gene. It is controlled by the gene, and we have many genes, right? For every function, there are genes, different genes. Sometimes few genes combine and it will form one function, right? So we called it Operon, right? So when 2, 3 genes, 5 genes, 50 genes is combined together, right? And it's perform one function. We called it Operon. So we have many genes. How many functions we are having in the body? Thousands of, right? Thousands of functions, right? Hundreds of proteins are prepared every time, every day, right? So these protein preparation is controlled by what? This protein preparation is controlled by the gene. Now, genes regulation and expression, how these genes are going to be regulated and how their expression can be read. So their expression is translated, if you remember in the previous lesson, you have studied their expression is translated from the DNA which is present in the nucleus, right? In the form of messenger RNA, right? And then it's translated to protein, right? So that is actually what? That is actually the gene expression. Now how they are regulating, right? So gene expression and regulation. If you look to the cancer cell, right? Cancer cells, how they are producing abnormal growth, right? Uncontrolled growth. When the cell are growing by itself, so it will cause what? It will cause cancer, right? So if there is no regulation, right? The cell is not regulated by the genes. So what will happen? It will cause a lot of disease. Like we know a disease, cancer, which is the most dangerous disease, right? It's occurred due to what? It's occurred due to the irregulation or uncontrolling of the genes. The genes are uncontrolled. To be specific, there are protein which is responsible for the cancer cell. Gene P53 is responsible, but the gene P53 is preparing a protein which is called regulatory protein. Which regulatory protein? Pectin, I think it have, or like this, it has a name, pectin or protein, but it's a regulatory protein. It is a group of protein which regulate the function of cell division, right? In chapter 10, we have, when we studied the cell division, so there we studied a protein which is regulated by P53 gene, right? So that protein, when P53 gene is not working, so it prepare the protein, regulatory protein, and it's not working proper. So if it is not working proper, it leads to cancer. Okay. Now the lesson. Think of a library filled with a book, right? If you have a library, right? Let's suppose you will take your bag, right? Small library. You have the bag, right? You have 5, 6 books. Right? You are able to study these 6 books all in one day or one time or not? It's not possible, right? It's not possible. You will study only a page, a topic, right, or a few topics, right? So the same, if you look to the organism, right? It has many genes. It has many genes, thousands of genes in that. Right? Example, we have the word E. coli. Yes, for E. coli is a bacterium, right, which has around about 4,000 genes. Right? So 4,000 genes is not less, it is too much. In human, I think around about 40,000 genes are identified till now. When I was studying till that time, 40,000 genes were identified. Around about 5 years before. So 40,000 genes in the human is identified. So everyone is preparing different protein. The example which we have here, this is 4,000 gene, 4,200 I think 88 or something like this has. But we are taking 4,000 genes as it's in the bacteria. So all these 4,000 genes in the bacteria, it will not express at the same time. It will not translate into the protein into the same time, right? The same as like you have the book, and from the book you are doing what? You are taking just one lesson, one chapter, one topic. So the same it will happen there. Now leave the bottle, please. Most of the genes code for the protein that do everything from a building cells, cell wall to breaking down the food. Like for example, these 4,000 genes is responsible for the entire function of the body, for the E. coli, right? The entire function of the body of E. coli. But what will be the thing? The thing is that if let's suppose it's performing different function, so all the function is not going to be performed at the same time, right? Like for example, if you're eating, so with the eating you are not running, right? With the eating you are not running. So if you are eating, you are not running, right? But if you are eating, with the eating you are breathing, right? Your heart is beating. So a few works will occur together, but not all. Not all will occur. If you are eating, you are not fighting, right? So all things which occur together, right? Like 2-3 functions, right? 2-3 functions occur together. So there will be 2-3 genes which will perform. While we have thousands of genes in the body, hundreds of genes in the body, it will not work. Like let's suppose if you are eating and beside eating, what is the other process? Okay, let's suppose take digestive system. If you take digestive system, so in digestive system, many events occur when you take food into the mouth, right? So first, movement occur in the hands, right? So this is controlled by what? The genes which is present in the nervous system, right? It is controlled. Now you take food into mouth, right? In the mouth, again movement occur. Movement of jaws occur. After movement of jaws, another process will occur, which is the secretion of amylase, which is the secretion of saliva, right? By which the food are getting soft. So this is another gene involved, which is from the endocrine system, right? On from that, the food will go to the stomach. So in the stomach, we have different juices, digestive juices, like HCl. So HCl is secreted by another gene. This actually what? HCl are enzyme. An enzyme or what? It's protein, right? If it goes down to further part, like intestine, so in the small intestine, different juices are performing function. So in the same digestive system, we have many genes involved, many functions involved, many proteins are involved. In the same digestive system, taking right from the food, even before the food you are eating, right? Before you are eating the food, you come to school at 7 o'clock, right? When you come to 7 o'clock, you're not eating till 10 o'clock or 11 o'clock till the break, right? So before the break, 20 minutes, 30 minutes, or 1 hour, what generate it? This now 4 o'clock, I didn't, 4 hours, I didn't eat anything. So now your brain signals, right? The gene expression start in your brain that now the stomach is getting empty, right? So the stomach will produce what? The stomach will produce digestive juices which will produce appetite, right? So then what you are thinking? You are just walking toward food. And when you eat the food, it's what happened? Then the genes stop the expression. So when genes stop the expression, so it means that you fulfill the need, right? Like let's suppose you are walking in the street. You are walking in the street and there is a dog. What will happen, right? If the dog runs toward you, so you will also run, right? So this is response, right? So this all is being regulated by what? These all are being regulated by the genes. Now, prokaryote, we have 2 types of cell, right? Or 2 types of organism. One we have eukaryotes and other is prokaryote. What is eukaryote? Eukaryotic cell. Eukaryotic cell. Who know eukaryote and prokaryote cell? I don't think. Eukaryotic cell and prokaryotic cell.
Prokaryotic cell is the most multicellular organisms, and it has a nucleus. The DNA is inside it.
Yes, that is the point, right? If you look to the prokaryotic cell, eukaryotic cell, they have nucleus, EU, which contain nucleus, and prokaryotic cell, those cells which do not have nucleus, right? So if they do not have nucleus, so the nuclear material it will be present in the cytoplasm, right? Scattered in the cytoplasm, and it will have one circular DNA. It will have what? It will have one circular DNA. So 2 type of cell we have. One we have prokaryotes, other we have eukaryote, right? So the first one, which is the simple one, that will occur in, that is prokaryote, right? Which do not have nucleus, or we can say membrane bonded nucleus and membrane bonded organelle. How are prokaryote gene regulated? How the gene regulation occur in the prokaryote, right? So gene is present where?
the cell, in the nucleus.
Cell, nucleus, inside nucleus, DNA, right? DNA or chromosomes. You can just say both of them. DNA or chromosome. So if there is no nucleus, so we are saying that in the terms of prokaryote, that the single circular DNA in the prokaryote will be present scattered in the cytoplasm. So if there is no nucleus, we will check for the gene, right? So in the prokaryote, the gene will be present on the DNA which is present in the cytoplasm. DNA binding protein in the prokaryote regulate gene by controlling transcription, right? Transcription is the process by which the short sequence of the DNA transcribed to messenger RNA and then translated into the protein. This process is called what? DNA binding protein. Those proteins which are going to bind to the DNA, right? DNA binding protein in prokaryotes regulate gene by controlling transcription. So the protein which bind to the DNA in the prokaryote, they are going to control or they are going to regulate the gene expression in the prokaryote. Bacteria and other prokaryote. Bacteria is the common prokaryote, right? So that's why we are taking first example on bacteria. Bacteria and other prokaryotes do not need to transcribe all of their gene at the same time, right? As I told you, that let's suppose you have the book, you have the bag. So in the bag you have thousands of pages, right? Each book contains, right? So you're not taking all of them at one time or one day. Just one page, 2 page, or 3 page, right? So the same it happens in the case of the organism. Like bacteria, which we take the example, E. coli have around about 4,000 genes, right? So around about that 4,000 genes, all of them are not performing function, right? Some of them which are active, they are going to perform function, and function mean they are going to express. Gene expression will occur. Gene expression is function, right? Gene expression is the word. Gene expression is used for functioning, right? The function of the gene. To conserve energy and resources, prokaryotes regulate their activities, producing only those genes necessary for the cell to function. Only those genes will work which is necessary for the function, right? But another point here it is that let's suppose 100 genes out of these 4,000 genes are functional, right? It's performing function. Still 10 out of these 100, let's suppose it will be activated at one time, right? Let's suppose the bacteria is in the reproduction mood. So only these genes which are responsible for the cell division, for the reproduction, they will be activated, right? Or let's suppose it is the feeding time, eating time. So those genes which are responsible for the food, they will be activated, right? Let's suppose it is in the movement. So those genes which are responsible for movement, only they will be activated, right? For example, it would be useful for the bacteria to produce enzymes that are needed to make molecules that readily available from its environment. So in the case of like food, right? And what will happen in the environment? In the environment, bacteria will look for food mostly, right? By regulating gene expression, bacteria can respond to change in their environment. The presence or absence of nutrient, for example, right? So bacteria is always what they will look for in the environment for food. So they will sense or they will feel the environment, whether there are food or whether there are not food. So if food, it will check the food. If not food, so they will search for food. Then motion or movement will occur. Some of these regulatory protein help switch on while other turn gene on, right? So switch off and switch on mechanism, right? Feedback mechanism. Have you studied it before or not? Feedback mechanism. Feedback. Okay. So let's suppose switch on and switch off, right? As the switch is working for light, you are going to turn it on and you are going to turn it off, right? So the same genes are working to turn it on and turn it off. The example which I give you for the appetite, like for example, as I told you that you come 7 o'clock to the class, right? 4 hours you are sitting here, right? After 4 hours, after 3 hours, you are feeling that I need to eat something, right? So this is actually what now the gene which is responsible for the production of digestive juices, it is being turned on, right? And it is producing digestive juices in your stomach, and you are feeling that now I need to eat something. When you eat food, right? So what happened? The gene which is going to secrete the digestive juices, right? Which is responsible for the stimulation of the digestive juices. So their gene turn off again. So when the gene turn off again, so it means that the production of the digestive juices stop. So when the production of the juices stop, right? So when the gene turn off, so production will stop. It means that you have eaten something. Right? So turn on and turn off mechanism, right? Another example which I give you about, let's suppose you are walking in the street and there is a dog, right? And it's running toward you, what will happen? You will also run. But when you reach to a safe place, so you will stop, right? So that is what? They are genes which is in your mind that run, right? But when you reach to a safe place, so it turn off. That now there is no danger, so you can stop. So turn on and turn off mechanism, right? It is also important. Gene in the bacteria are organized into operon, right? As I told you that if you look some genes which are responsible for a function, let's suppose 2 genes, 3 genes, 5 genes, 10 genes, 50 genes, when it is responsible for a specific function. For a specific function, group of genes, we call it operon, right? Or sometime, not always, sometime they are also counting for closely related function. Right? Like let's suppose, to be specific, we will take any part of the digestive system or any system. Let's suppose we will take the stomach, right? So what happened in the stomach? Food are going to digest with the help of enzyme. 2 things, right? Amylase and HCl, right? Amylase comes from the mouth along with the food, and HCl it will be secreted by the stomach, right? Yes, into the stomach. So these two digestive juices, amylase and HCl, right, hydrochloric acid, which is. So both of them, they are acting on the food. So now the genes which is responsible for controlling these two, right? So sometime we are also going to count it into operon, right? But mostly it will be for one function. Like example, they give you about with gene. An operon is a group of genes that are regulated together. The gene in an operon usually have related function. Related function or one function, right? So if it is like in the one system, mostly in the one system it is related function, right? Like immune system. The genes are the proteins in immune system. What is its function? Healing process, right? Or fighting against diseases inside the body. So mostly these are interrelated function. Let's suppose heart beating or blood circulation, right? These are what? These are related function, right? So the gene which is responsible for related function, they are going to be counted as what? They are going to be counted as operon. Okay, example you have here for leg operon. Okay, leg operon is actually what? If you look to the name leg operon, right? Leg. So the word leg is being taken from lactose, and lactose is a type of sugar, right? Lactose is a type of sugar. Leg is being taken from lactose, right? Lactose. So leg operon. Leg operon is what? If you look to the bacteria, which we have example for discussion, E. coli. So E. coli has 4,288 genes, right? 4,000 we are taking. For example, the 4,288 genes that code for protein in E. coli include a cluster of 3 genes that must be turned on together before the bacterium can use the sugar lactose as a food. When you suppose bacteria sense, right, or it touch any sugar in the environment or in nutrients, right, and it is what? It is lactose. Sugar has many types, right? So lactose is one type. When lactose is being sensed in the environment by the bacteria, so what will happen? The lactoperon will turn on. Lactoperon contains 3 genes, right? So there 3 genes, all of the 3 genes which include in the lactoperon, they will turn on. Why they will turn on? Because lactoperon is responsible for the digestion, or you can say for the digestion of the lactose, which is sugar. So that's why these 3 genes collectively it's called lactoperon. You understand? Did you understand or not? What is leg operon? Who can tell me now? Okay, simple. What is operon?
genes are related to growth. Group of genes that are related together.
Yes, a group of genes that are going to perform the specific function are most closely related function, right? Okay. The leucoperon, lactose is a compound made up of two simple sugar, galactose and glucose, right? So simple, you can say it is what? It is a type of sugar, right? A type of sugar molecule, which is made from galactose and glucose. To use lactose for food, the bacteria must transport lactose across its cell membrane and then break them the bond between glucose and lactose. These tasks are performed by the protein coded for the genes of leucoperon, right? So if the bacteria is going to digest or break down the galactose or glucose, right? When two simple sugar combine, it form what? It form lactose, right? A type of sugar. So when lactose, a type of sugar, is going to digest by what? It's going to digest by the bacteria. So for this, the responsible operon or the responsible gene are what? It is leucoperon, right? So this food particle, which is lactose, it needs to be transported inside the body. So once it is transported inside the body of the bacteria, so which gene will turn on? Or which operon will turn on? The leucoperon will turn on or switch on, and it will perform the function of the digestion. If the bacteria grows in a medium where lactose is the only food source, it must transcribe these genes and produce these proteins. So the same part, if all the food is lactose, so all the genes, they will work, or you can say that the leucoperon, it will be only turn on, switch on, and it will perform the function, it will produce these protein. If grown on another food source such as glucose, it would have no need of these protein. The late genes are turned off by the protein that bind to the DNA and block the transcription. Let's suppose there is another, the bacteria is living in the place where there are only lactose, right? Lactose is only the food bacteria. So then the leucoperon or the will be activated and it will transcribe the protein, right? But let's suppose the bacteria is present in the food or in the place where there are no food related to, the food didn't contain any lactose sugar, right? So then what will happen? Then the leucoperon will remain off, right? It will switch off always. Then other operon or other genes which is responsible for the food which is present here, so it will turn on. Okay, if you have any question, you can ask me now. We will stop here. Then the topic is going to change. If you have question, you can ask me. Yes, this is any. This will be a little bit difficult to have, right? So, this is also included in the exam. So you have to keep it in mind. If you have any question, you can ask me.
عبود احمد عمار احمد محمد طه الصوت هذا الجاب اجلس شوي عمار احمد محمد طه اهلا بالله زهراني فيصل ناصر محمد فاك صلاح محمد جمال احمد اسمه الباني وبعد وكمل يعني